Hi everybody, and welcome to our module on DNA mutations. Before we get into DNA mutations, I want to briefly review a few very basic concepts about how proteins in the body are synthesized. So the blueprint for proteins in our body is found in our DNA, and via the process of transcription, DNA is transcribed into RNA, and then via the process of translation, RNA is converted into proteins. Codons are three nucleotide sequences found in DNA and RNA, and they are the language that our cells use to create proteins. So for example, the three nucleotides TAC, if found in DNA, would be transcribed into AUG in RNA, and then via translation, the AUG codon, these three nucleotides, codes for the amino acid methionine, and that methionine would be incorporated into a growing protein based on the codon found in DNA and RNA. Shown on the screen is the genetic code that cells in the human body use. It shows the three nucleotides you can find in RNA and which amino acid they code for. So for example, a UUU, if found in an mRNA molecule, would code for the amino acid phenylalanine. An AAA would code for the amino acid lysine, and so forth. You'll also notice that there are three stop codons here, UAA, UAG, and also UGA. If these three nucleotides are encountered in mRNA, that tells the body to stop synthesizing the particular protein that it is making. Now that we've reviewed those very basic concepts, we can begin our discussion of DNA mutations. DNA mutations are error in DNA, and they can be a simple mutation that involves only one or a few bases or nucleotides that become abnormal in a particular stretch of DNA, or they can be complex that involve entire gene deletions or translocations of entire genes from one chromosome to another. One way to classify DNA mutations is as germline mutations or somatic mutations. Germline mutations are mutations that involve the DNA of sperm and egg cells. These are gametes, and therefore the mutation can be transmitted to offspring, and when they are, the DNA mutation will be found in every cell in the body of the offspring, including the germ cells of the offspring, so that the offspring can then pass it on to subsequent generations. So once a DNA mutation involves the germline cells, it can be passed down through a family. In contrast, a somatic mutation is a mutation acquired during the lifespan of a cell. For example, if one of your skin cells develops a mutation, that skin cell can pass it on to daughter skin cells, but it's not going to be transmitted to the offspring of the individual because it doesn't involve the germline cells. The first type of DNA mutation we're going to discuss is called a point mutation. This is where a single base is switched with another base, swapping the nucleotide from the correct nucleotide to the incorrect nucleotide. There are two subtypes of point mutations. Some are called transitions. This is the more common type. Others are called transversions. A transition involves a purine to purine swap or a pyrimidine to pyrimidine swap. So for example, if you look at the right side of the screen, cytosine and thymine are the two pyrimidine bases. They both have a single ring. Because they both have a single ring, they often fit in the same space as the other, and therefore these types of point mutations are more common. Also at the bottom of the screen are the two purines, adenine and guanine. They both have two rings, so once again, they often fit into the space of the other, and this results in a transition point mutation. Less common is a transversion, where a purine is swapped with a pyrimidine or a pyrimidine with a purine. This is less common because of the different sizes of the bases. Because of a phenomenon in our genetic code called Wabble, some transition point mutations are less likely to alter amino acids in the resulting protein. The reason for this is because our genetic code often codes for the same amino acid with an altered base in the third position. For example, let's imagine that we have a piece of RNA with a UUU, and there is a transition point mutation, and the third U becomes a C. This is a transition because we're swapping one pyrimidine for another. This transition, however, will not change the resultant amino acid because both UUU and UUC code for phenylalanine. In other words, UU and any pyrimidine will code for the same amino acid. This phenomenon is called Wabble, and because of this, some transitions are less likely to alter the resultant amino acid and change the protein that results from that mutated piece of DNA. We can classify point mutations in a number of different ways. One way is as a silent point mutation. This happens when the nucleotide substitution codes for the same amino acid. This is what we were just talking about in the last slide. Because of Wabble, this often happens when there's a base change in the third position of a codon. So for example, if your DNA had AAA, this would create a piece of mRNA that was UUU, which codes for the amino acid phenylalanine. If that third A were switched and became a G, this would make a piece of mRNA that was UUC, and that still codes for phenylalanine. Therefore, this is called a silent point mutation. It has not resulted in any change in the amino acid or the protein that is synthesized. Another type of point mutation is called a nonsense mutation. This is a nucleotide substitution that results in an early stop codon. As we said before, stop codons are nucleotide triplets that signal the termination of translation of proteins, and there are three stop codons: UGA, UAA, and UAG. So for example, imagine that our DNA was ACC and that coded for a piece of mRNA that was UGG. This would code for the amino acid tryptophan. If that last position, the C, were switched to a T, this would now code for UGA, which is a stop codon, and as a result we would get a truncated protein because of this mutation. A final type of point mutation is called a missense mutation. This is a nucleotide substitution that results in a different amino acid in the final protein. For example, imagine if we had this stretch of DNA that coded for this stretch of mRNA. This would synthesize a protein with 4 histidine amino acids. If we change just one nucleotide, this T right here, to a G, we would then get this stretch of mRNA, which now codes for a histidine followed by a proline followed by 2 histidines. This is called a missense mutation. We have changed the amino acid in the final protein because of a point mutation. The classic condition caused by a missense mutation is sickle cell anemia. The root cause of sickle cell anemia is a missense mutation in the beta globin gene. Sickle cell anemia is caused by a single base substitution in the 6th codon of the beta globin gene. An adenine base is changed with thymine. This results in an amino acid change where valine is substituted for glutamate in the beta chains. On the bottom left side of the screen, I've shown you the makeup of someone with normal beta globin genes. There's a double strand of DNA, and the template strand that is used to make RNA is CTC. This codes for an RNA molecule that is GAG, and that codes for glutamate, and thus glutamate is placed in the beta globin chain. On the right side of the screen, I've shown you the situation in someone with sickle cell anemia. The template strand of DNA has adenine substituted for thymine. This leads to an mRNA molecule that is GUG, and that codes for valine, so the amino acid valine instead is inserted into the beta globin gene, and that leads to all the clinical consequences of sickle cell anemia. Now we're going to move on from point mutations and talk about insertions and deletions. So in a point mutation, one nucleotide is swapped for another, but the size of our DNA does not change. In an insertion or a deletion mutation, there's addition or subtraction of nucleotides, and this alters the size of our DNA. And just like a point mutation, this can alter the protein product of a gene. And there are many examples of insertions and deletions in genes that cause disease, but one classic example is cystic fibrosis. The most common mutation is called the delta 508 mutation. This is caused by a deletion of 3 DNA bases or nucleotides that leads to the loss of the amino acid phenylalanine in the final protein product. This leads to abnormal protein folding, and that's what causes the clinical condition of cystic fibrosis. Some insertions or deletions lead to what's known as a frameshift mutation. This is an insertion or deletion that alters the reading frame of our genetic code and has a major impact on the final protein product. To understand frameshift mutations, let's look at this drawing I've made on the screen. At the top is an example of a point mutation. We have a stretch of DNA that codes for 4 histidine amino acids. We then change this single T right here to a C, and we now get another stretch of 4 amino acids. The first one and the last 2 are unchanged, but the second one is changed to an arginine. What's happened here is that we have not changed the reading frame. This is a reading frame, that's one codon, this is the second codon, this is the third, and this is the fourth. And even though there's been a mutation, the first 3 are still being read as one codon, and the last 6 are still being read as a codon. This is called maintaining the reading frame. At the bottom, I've shown a frameshift mutation. We have the same stretch of DNA that codes for 4 histidines, and then we have inserted an A into this DNA sequence. This has now altered the reading frame. No longer are the GTA the first codon. Instead, the first codon is AGT, which codes for serine. We then have another 3 serines. So because of this single base insertion into DNA, we've altered the reading frame and made a massive change in the resultant protein, much greater than the single amino acid change we made at the top. So a frameshift mutation is caused by a deletion or insertion that is not a multiple of 3. This leads to misreading of the nucleotides and codons downstream from the mutation, and this can result in a significant change to the final protein. As we saw in the last slide, many amino acids can be changed by a frameshift mutation. In addition, the frameshift can result in an early stop codon, which leads to a truncated protein, or it can result in loss of a stop codon, which leads to an elongated protein. Frameshift mutations have been described in a number of genes that lead to disease. This type of mutation has been described in Tay-Sachs disease, which is a lysosomal storage disease. Frameshift mutations caused by insertions and deletions have been described in the gene for hexosaminidose A that causes Tay-Sachs disease. In addition, Duchenne muscular dystrophy is a classic example of a frameshift mutation that causes disease. This condition is caused by a frameshift mutation in the dystrophin gene that leads to absence of functional dystrophin. And in contrast, Becker muscular dystrophy is a much milder version of muscular dystrophy, and the reason it's milder is because the mutation in that condition is not a frameshift mutation. The final topic for this video is slip strand mispairing, or DNA slippage. This is a phenomena that occurs in segments of our genome with repeated nucleotide sequences. It can be prevented by mismatch repair mechanisms, but if those are inadequate, this can lead to DNA slippage. And DNA slippage can cause insertions or deletions, which, as we discussed before, can potentially lead to frameshift mutations. In order to understand this phenomena, you need to appreciate that in many stretches of our genome, there are nucleotide segments that are repeated and that are close together. In other words, they look just alike and they are near one another in our genes. For example, imagine that we have the two complementary strands of DNA shown on the screen. The top strand has two A's paired with two T's. There's then a short stretch of nucleotides represented by the blue line, and then once again we have two A's paired with two T's. This is the type of portion of our genome where DNA slippage can occur, and there are many parts of our DNA where this type of thing is found. It's not always two nucleotides paired with two nucleotides as I've shown here. It may be 3 or 4 or many more, but it doesn't matter. The point is that there are nucleotide sequences that are identical to one another and that are close together in our genes, and that is the right setup for DNA slippage. To understand slip strand mispairing, let's imagine that we have two complementary strands of DNA with repeated nucleotide sequences, just like I showed on the last slide. Now let's imagine that these two A's mispair with the wrong T's, and we push these two A's out. This is going to result in potentially an insertion or deletion because of this mispairing. If these two strands I've shown on the screen become a template during DNA replication, this will result in a deletion. On the bottom strand, the full stretch of DNA will be replicated. On the top strand, this pair of A's will not be replicated and will get a shorter stretch of DNA than usual. If this slippage occurs as these two DNA strands are being replicated, then this top one may be replicated all the way out to here. Now we have a longer stretch of DNA on the top compared to the bottom, and this will have consequences when these two strands separate and replicate. This will result in an insertion. So the big point here is that because of this mispairing, you can get deletions or insertions in DNA, and this can result in variations in the length of our genome in portions where we have nucleotide repeat segments. This can also potentially cause a frameshift mutation, which can lead to all the consequences we described before for frameshift mutations. In a separate video, I discussed trinucleotide repeat disorders. These are diseases that result from genes with repeated trinucleotide units like CAG. The disease gene has extra repeats of the trinucleotide unit, and this is what causes disease. And one of the mechanisms by which these extra repeats are believed to develop is from DNA slippage. And there are a number of examples of trinucleotide repeat disorders shown on the screen here, and I discussed these all in a separate video. In the colon cancer video in the GI section, I talk about microsatellite instability. Microsatellites are short segments of DNA that contain repeated sequences like CACA. If mismatch repair enzymes fail, this leads to instability of these segments, and by instability, I mean variation or instability in the size of the segments among cells, and this is seen in colon cancers. So one colon cancer cell will have a long stretch of a microsatellite, another colon cancer cell will have a short stretch, and another one will have a stretch somewhere in the middle. This is called instability of these microsatellites, and one of the mechanisms by which these develop is DNA slippage in the setting of a failed mismatch repair enzyme system. And this is seen in the setting of colon cancer. And that concludes our video on DNA mutations.